everybody. Welcome to today's event. I am Ayush Panda, today's moderator, and I am joined by Yunis Bare, who will be moderating the event with me. Today's event is special. It is the first edition of our newly revamped series of talks called EV Isolation and Characterization Series. This series of talks is dedicated to exploring the new developments in technology for efficiently isolating and characterizing EVs. And today, we are joined by Professor Joshua Leonard, and we couldn't have asked for a better speaker to launch this series. Professor Leonard trained in chemical engineering, receiving his bachelor's from Stanford University and a PhD from the University of California, Berkeley, where he worked with gene therapy pioneer David Schaffer and earned a certificate in the management of technology from the Haas School of Business. He then trained as a postdoctoral fellow at the National Cancer Institute, where he elucidated features of the innate immune system that drive antiviral immunity and inform vaccine design. He then joined the faculty of Northwestern University in chemical and biological engineering. His lab engineers novel biological systems that perform customized, sophisticated functions for applications in biotechnology and medicine, helping to build the now vibrant field of mammalian synthetic biology, employing methods ranging from biomolecular engineering to computation driven design. His team develops technologies including programmable cell based devices for treating chronic diseases, including synthetic receptors and genetic programs, and novel gene therapy platforms based upon bioengineered nanoscale vesicles. Professor Leonard is actively engaged in the development of national science policy, testifying as an expert witness before the US House of Representatives on 21st century biology, and through his role as a council and board member of the Engineering Biology Research Consortium. He fosters training, entrepreneurship, and industrial impact as director of Northwestern's NIH funded biotechnology training program and entrepreneurial activity as a founder and chief scientific officer of Synex, a startup focused on making transformative gene delivery technologies universally accessible. Professor Leonard is a founding member of Northwestern's Center for Synthetic Biology, which has grown to be a leading team within the SynBio community. Today, Professor Leonard will present work his lab has led to characterize and ultimately modulate EV-mediated delivery of biomolecules. I request the audience to put their questions in the chat box, or you can just unmute yourself and ask the questions at the end of the talk. Now, Professor Leonard, the stage is all yours.
Great, thanks so much. Are you seeing my slides okay?
Yes, perfect.
Okay, fantastic. Well, thank you for that very lovely introduction, and it's a real pleasure to be here. I'll try to actually connect some of those background pieces to the talk today because, I think in general, understanding why we do the things we do is really helpful to understand where the opportunities are and maybe where the next steps might be. So that's going to be a theme I'll use throughout the talk today. And in fact, because the focus of my lab works on synthetic biology generally, I'm going to connect that broader focus of what we do to the specific facets that go to EV bioengineering and talk about how we as engineers think about the challenges of bioengineering EVs for functional applications, but also for the characterization opportunities that are the real theme of the sequence here. So that'll be the overall arc of the story today. And I brief disclosure here on the startup company here. This is something I'm going to weave in actually to the story here because I think it'll be interesting to think about how the stuff we do in the lab has an impact in the real world, connects directly to the story we'll be talking about today. So stay tuned for that. Before we do, I'll just zoom out a little bit. And for the sake of the audience here, who's going to be coming from a lot of different places here, I'll talk a little bit about what we mean by synthetic biology within my group and really try to understand how that type of emerging engineering discipline fits into the broader landscape of all the things we try to do in bioengineering. And there are many facets of synthetic biology as a field, ranging from microbial engineering through development of new therapeutics. But one concept that unites all these efforts is really trying to understand how to make the engineering of biology more of a mature engineering discipline. And so that doesn't mean necessarily knowing how everything works. In fact, that's not how engineering works in any way. What you do is try to make progress, and you try to make progress by having the ability to go from a goal you're trying to address to having a thing you can build to testing that prototype and then be able to improve it over time. And the things that make that go faster in all the mature engineering spaces we have are things like quantitation, computational analysis, and modeling, certainly very aligned with, I think, the objectives of the talk that we are having here today. And my group has been working on that for a number of years, really anchored primarily in the goal of trying to make engineered cell therapies. And again, back to the story about my training coming from the NIH, the real learning from that was, how do clinicians and immunologists come up with great ideas that if we could only build something that manifested that idea, we have a shot at coming up with a better therapeutic. And so my job, I think, as an engineer, our job as a team here is to make that process faster, go from the idea to the thing we can test in a clinical trial, for example, faster, cheaper, more efficient, and ultimately making medicines more accessible. So that's overall what we try to do within my group here. And so a lot of that comes down to, at its basics, a lot of biomolecular engineering, right? So if we think about a cellular therapy as a cell, you would say a receptor, a signal transduction cascade, gene regulatory bits, but there's actually a lot of merit in thinking about those same processes as parts, sensors, processors, and actuators, in part because that's the way we can take a complex goal and break it down into a series of individual pieces that let you actually build a thing that you can then test out your idea. So in work I won't talk about today, we've done a number of projects building synthetic sensors, synthetic gene regulation programs, synthetic actuators, and have got pretty far to the point, I think, where as a field, not just my group, of course, we can do things like this illustration is suggesting, which is do kind of CAD-based simulations of things that might work to guide what we build so that they are more likely to work before we do any work in the laboratory. This is kind of in very much alignment with the AI and machine learning revolution of especially things like DNA and protein engineering, where you can see this is now something that people can do at the molecular scale. And I think we can increasingly do this at the system scale, which is really what this cartoon is meant to address. So all that's to tee up. This is what we've been doing since I started my lab here. And this is a big part of what got interested in EVs right around the time that some very exciting things happened in the EV field. So I'm going to talk a little bit about that history here, which is probably something you all know about, but I want to contextualize what we've done within that revolution in the EV space. So I've been interested in these actually since I was a postdoc. The lab next to me was studying the way that EVs play a role in antigen cross-presentation. Seemed like a really cool scientific thing that was really emerging. That would be like in the late 2010s, something like that, right? 2005, 2010. And so, of course, the field of EVs goes back to about the 80s when these things were first characterized. And there was a transformation in the way we think about EVs that I think is useful for thinking about where we've come even in the last 10 years. And so I'm going to indulge myself with a little bit of a summary of this through the lens of this really great kind of conceptual framing of the structure of scientific revolutions. It's a book worth a read, but it basically is how whenever ideas change, there are these moments of foment where it's not clear what's going on before it kind of crystallizes the paths forward. And so I'll talk about, of course, there are many versions of that story you can tell within the EV space, but I'll talk about the application-oriented view that got us interested. So, of course, we knew these particles had existed since about the mid-20th century. Around the late 90s, it was shown that these can play a role in playing materials with some really pioneering ideas about harnessing that for making vaccines. Eventually, the role in cancer became more and more established. And around this time, I would say, the idea that these EVs can load and transfer nucleic acids, modulate the function of recipient cells, really piqued a lot of interest in terms of thinking about these as not just a neat part of biology, but something that we could use in an application-oriented way. And of course, there was this kind of like gigantic explosion then of like, is everything mediated by EVs? And it turns out like many things, there is a role for EVs, which is a big part of course, why we're all meeting together in this talk today. The reason we got interested around that same period was primarily thinking about this as an application. And so if you imagine the type of cell therapies we were trying to build, oftentimes we talk about the control of what the cell does, but then what does it do when you have executed the sensing and information processing steps? You can engineer the cell to make a protein. You can engineer the cell to turn on a native regulatory function. And EVs are potentially a way that your engineered cell can act upon the rest of the body in ways that are ultimately maybe inspired by the way we know the EVs work in real life, right? And so that's where we got involved, and specifically thinking about EVs as a facet of the kind of transport systems that my group have been studying for a long time, which is really thinking about viruses as a model that have been used to develop virus-like particles or virus-derived vectors. That's similar to this concept of thinking about, look at how things have naturally evolved, how can we harness those for useful purposes? And it's usually not right off the shelf, but it tells you there's a opportunity there that could be exciting. And so a big, I think, spark for many of us was this really groundbreaking work by Matthew Wood and team, where they showed in this neat, very, very early study that there was some potential to harness EVs for doing some kind of therapeutic gene delivery. And it was early days, and so there were a lot of provocative concepts raised at that time, where maybe these properties are distinct from what we can do with synthetic particles in terms of being low immunogenicity, low toxicity, and maybe having unique transport properties within the body here. And that really kicked off a big I would say, explosion of work trying to think about using EVs for applications. And so I'll just kind of summarize a little bit of that thinking maybe in the intervening years, but that's where we jumped in and really thinking about EVs as these promising genetically programmable multifunctional nanoscale vesicles. That's the way we think about it, right? So in terms of looking at the heterogeneity of EVs, you can imagine two perspectives. You can look at what EVs have, which is a big part of the characterization bit, but you can also look at what EVs can have, which is thinking about how can you deliberately cause them to have certain features and have certain functions, and that what can EVs do perspective is what's guided a lot of the work that we've done over a number of years, really as a way of interrogating aspects of EV biology as well. And by we, I mean the field in many ways. By trying to enhance loading, you can identify when is it limiting, when is it not limiting. By trying to enhance targeting, you can detect what the limits are and so on. And finally, delivery. So I would say that over the period of time, I've got a couple of papers here that I won't be talking about, but we did, I'd say, individual contributions along the way for these active loading bits, looking at targeting, oftentimes finding those limits as part of those studies. Really, really informative for thinking about, okay, what do you need to do to put all these pieces together and have a complete functional delivery platform there, which had been fairly elusive in a lot of different applications. I think that was a big, goal among us in the field more generally. And there were a lot of promising observations, but it was still kind of unclear how to put all these pieces together. And so what's been really exciting in the last couple of years is there's been a series of breakthroughs, I think, to really show how this perspective of trying to probe what EVs can do can actually bear fruits in terms of functional outcomes. And so I'm going to just tell you two stories today that kind of picks up on this thread. One is a recent paper where from my group where we showed how if you can figure out how to put these pieces together, you can do a functional delivery application. So this application-driven view will kind of tee up what will then become the questions that were raised and really necessitate characterization, and that's where we'll end up for the rest of the talk today. Okay. And again, we can come back to any of these in questions if you all have follow-ups you'd like to pose. So part one is this application-driven perspective. How does this prepare us to use EVs, and how does this raise questions about what comes next? Okay. All right. So in this slide, I'm going to move this here. In this slide, what we've done is we've set up a goal statement for this paper that was published in last year in Nature Biomedical Engineering, where we were trying to, in the sort of narrative, we were trying to figure out how to deliver Cas9 ribonucleoproteins to primary human T cells in order to edit the locus that encodes for the HIV co-receptor CXCR4. And so that was the motivating application, but as much as any direct utility, that was also a really hard set of things to do because you have to put all these pieces together. And that was really good for us for tech development because, for example, one thing we and others in the field had learned is that the way that endocytosis affects EV deliver is very tricky to decouple sometimes. So a nice and challenging feature of T cells is they have very low endocytosis, which means it makes it hard to deliver, but also easy to figure out if you're making a difference in terms of the delivery efficiency. So that was a good motivating application for technology development. And then, of course, figuring out how to test whether this had been a true functional delivery of contents of EVs made us attracted to things like Cas9 delivery, since you can tell when there's a DNA edit if the cargo was truly released versus just trapped in an endosome somewhere, which is oftentimes a challenging thing to separate. Okay, so I'll just mention a few things that we did in this study, highlight a couple key points, again, characterizing the things that are interesting and really questions we wish we could have answered from a characterization standpoint in this paper. Okay, so I'll just walk you through kind of how in general we broke this down. It's really similar to the setup I had mentioned with looking at loading, targeting, and functional delivery. So targeting is the one I'll tell first in this story here. At the time we started this, there wasn't a demonstrated way to put affinity reagents beyond peptides on the surface of EVs. And so we explored a number of just protein bioengineering strategies for putting SCFEs on the surface of T cells. We started with an anti-CD2 single-chain antibody, in part because for the HIV treatment application, this CD2 is a putative marker of the HIV, the T cell pools where HIVs may reside in latent HIV infections. That was the big motivation for picking that particular affinity reagent to drive this tech development. And so we can do things like make a bunch of different versions of proteins that could encode that SCFE. And I'm showing you here kind of like what a couple of years really of tech development led us through, where once we developed the assays, you can see with using fluorescently labeled EVs. And so in this case, we're using EVs that were incorporating a fluorescent protein cargo. Having tried dyes and other methodologies, we really landed on this protein incorporation as a really unambiguous way of quantifying average, let's say, EV uptake by recipient cells. And you can see in this assay where we're delivering to jerk-cat human T cells, non-targeted EVs give you no signal. The first couple of designs with this SCFV2 were promising in the sense that we saw delivery with both kinds of vesicle subsets. At this point, I should mention the caveat that when I say microvesicle and exosome, those are defined in terms of the gradient centrifugation that separates those pools. So in the paper, we are careful about using that sort of methodological definition here rather than biogenesis route. It's here as a shorthand for the rest of the talk today. That'll be true for everything I show you today. Okay. High-speed EVs, ultracentrifuge EVs would be another way people refer to those subsets occasionally. In any event, we see there was some degree of targeting achieved with this initial things, and then we did a lot of work to figure out how protein sequence changes, the hinge, the scaffold, all the things that go into proper folding as well as trafficking on the surface of a vesicle led us to get some much improved delivery at the end of that exercise here. So this just shows that don't give up if it first doesn't look very strong. There is oftentimes a lot of room for improvement, and it really, really, really helps to have a good negative when you're starting here. A challenge with trying this in other systems is if you get a lot of background, it's really hard to actually figure out when you're making things better, and that was crucial for this story. Okay, so once you do that, you can do sort of classic biochemistry tests. So for example, we proved that that interaction was mediated by the antibody, not just by leaving it in or out, because that changes all kinds of things about EV function. This assay is designed so you take one batch of EVs and you dump it on cells under two conditions, one where you've pre-blocked the target with an antibody and one where you haven't. And that kind of competition assay proves, in this case, that the interactions were specific to the antibody target, rather than, for example, making the EVs more sticky or easier to be endocytosed for reasons not attributable to the antibody. And once you've kind of honed in on that, it was nice that this data generated on T cell lines were replicated in primary human T cells. More or less the same pattern was observed. Although, frankly, it worked a lot better on primary human T cells than it had worked on the T cell line we had used. And that is kind of a theme that's worth some thinking about, too, across a number of papers, too. So this was positive. We were able to get targeting. The targeting is specific. And there was maybe a little bit of evidence that there was internalization happening as well, which we probed by using a trypsinization assay. Kind of modest, not really conclusive, I would say. And so that led us to say we need to really have a functional delivery assay to confirm that. And so the two steps of that are trying to figure out how to get the Cas9 into the EVs, and then you can measure the editing in the target cells. And at the time, there were a lot of different ways of doing some types of loading of cargo in EVs. But for this kind of an assay, it needs to be a kind of special balance of being able to get into the EV, but then be released from the EV remnants once it fuses with the target cell. And so there wasn't a thing that looked like it was perfect for us, maybe accepting some optogenetic methodologies, which are really nice. We decided to try an approach that didn't rely upon optogenetics for sort of pragmatic reasons. And so we explored the use of these small molecule-induced dimerization systems from plant proteins, in this case, abscisic acid, causes the dimerization of two proteins. And when you look at the whole cell, you can see that basically this was working as we wanted to. You add the small molecule, the fluorescent protein model cargo associates to membranes, and so it looked okay. this is a nice system to work with because the dimerization is reversible. If you wash out the small molecule, it dissociates, which is a contrast for things like rapamycin-induced dimerization domains, which are very slow to dissociate. So we thought it looked pretty good. We tested this out in a delivery experiment then. And so here's a kind of cartoon representation of all the different conditions where we loaded EVs with just cytosolic protein or with versions that have a combination of one half or the other half on either the fluorophore or on this membrane tethered antibody sequence here. And so you can see that the cytosolic loading is relatively inefficient. This is not a lowly expressed protein. Those cells are visibly fluorescent when you look on a microscope, very, very bright for whatever that's worth. But there's a lot of room for improvement when you add these active loading domains. The big shock for us was that it didn't work the way we thought it would, in the sense that having one half of the protein worked just as well as having both halves, and you didn't need the small molecule. So we decided that that was fortuitous, and we couldn't immediately explain it. And so we kind of put a pin in that and said, okay, we've discovered some kind of a spontaneous loading domain. Is it something that we can use? And so a test of that was, well, this clearly loads the fluorescent protein. Does the Cas9 get loaded, and is that Cas9 functional? And it was. We did a nucleic acid assay where we showed that if you made EVs loaded with Cas9 RNPs, you could cleave DNA in a specific way that showed that that Cas9 being loaded was active. So it's not aggregated or something else. So with that in pocket and a question mark remaining in our minds, we decided to move forward with that as a loading domain. And here's the final bit of that story. So we showed that when you put this all together and evaluate functional delivery, let's see what happens. First step is that we need to not only get taken up by cells, but escape. And so we looked at a couple of different ways of driving fusion. Sort of classic way of doing this is to express the vesicular stomatitis virus glycoprotein, or VSVG, on so-called vesicles, is another name used for these things. And those are fusogenic exosomes, or and microvesicles in this case. And we showed that if you look at just the uptake mediated by those glycoproteins, we see evidence of enhanced uptake with the addition of the VSVG. I'm saying uptake because this isn't really conclusive for delivery versus just inside of the cell here, although we washed for making sure it's not unbound. And we also explored the use of these measles virus HNF proteins, in part because these are shown to be pretty good at mediating delivery of, say, vectors to T cells. They're also pretty toxic, though. And so we kind of move forward with both of these evaluations here. Okay. This shows that those glycoproteins are doing basically what they should in this system. We move forward to a functional assay. So I'll just end this little arc here with this bit where we showed that EV delivery to primary T cells, dump the vesicles on, wait 6 days, extract the DNA, use NGS to measure the resulting edits here, that you get a distribution of edits that look like it should look if you're getting a cut with Cas9, like, say, delivered by an electroporation route. And so all these different spectra of edits are just important for making sure that when the, say, tag that we had and everything else is there, not only is it functional, but it seems to be doing this in a way where we're just looking at the diversity of ways of DNA repair that occur within the cell here. And so that was great. And we saw different levels of editing here with different kind of compositions, but I'll end the story with maybe the most interesting functional delivery data point. This is the last one from the story, where we tried doing things like repeated dosages. We did dosing with the microvesicle fraction as well as the exosome fraction. And we did this in a way where we tried to do a similar blocking experiment where we used CD2 binding antibodies to block uptake. Because, for example, if the binding of CD2 causes more endocytosis to be active in the cell, we wanted to capture that effect and basically control our interpretations that way. And what we found is that as you go to multiple dosages, you do get more edits, which is good. That's how it should work. In general, there were comparable, I'd say, degrees of editing for both the microvesicle and exosome fractions, maybe a little bit different, but there's some interesting things there. For whatever reason, there was a different, I'd say, degree of contribution of the CD2 domain. The way to interpret that is for the exosomes, for example, at these highest dosages, the blocking of that CD2 engagement really took a hit when you used EVs. For the microvesicles, it seemed less critical that the antibody be on the surface there. And so we can quantify all that stuff. And so the last thing we did then was, I think this is the first time I would say that we could do like one turn of the engineering crank, which is like, okay, this is what it is. Here's the baseline. Now, how do we think about the opportunities to improve? So we did a couple things that were actually suggested by reviewers. So a great idea. And so we did a couple of these things where we looked at just like how do these things compare to synthetic particles, so we could benchmark this to what other things we know about the literature. Altogether, that told us there's a couple things. One, there's probably room to improve the loading, the way we were doing it in that case with the ABI is probably not the only way, and so maybe some evidence for that. And finally, there's room to improve the degree of fusion that's occurring with those particles too. I'll say this: what inspired that was this measurement. We electroporated some EVs directly, some Cas9 RMPs directly into the cells with sort of a like maximal delivery efficiency, if you want. That's kind of our measure there. And if you did a control for like how many RMPs, how many molecules of Cas9 are we talking about here, we get about 1 or 2 logs more editing when you electroporate those RMPs directly into the cells compared to when you deliver them with EVs, which is actually a lot closer than we thought it was going to be, given that disruption by electroporation is probably the most efficient delivery you can do ex vivo. This looks like, though, there is some room for improvement if we could improve this. And so the end of the arc here, I think, is that this is a big like these final conclusions here are the first big, I'd say, forward-looking clues that once you have a system that you can use, you can make measurements, and then you can make all these conclusions about where you should point your next step of efforts. But I'll say a big part of this was there were all these questions we wanted to answer but couldn't answer at the time. For example, how many antibodies are there per vesicle? Great question. In the next part of the talk, I'll mention why we couldn't answer at the time, but I have some better clues about that today. Okay, so I'll come back to that in the second thing. Just to kind of close out this application perspective, we've done some work subsequently trying to improve some of these things. So one example is on this first topic, we teamed up with a colleague Neha Kamat here and published a story this year, I guess, looking at increasing or improving the way we can do loading of cargo using engineered protein lipid interactions. This is a really cool collaboration with, I guess I can say this for the first time, Nobel laureate David Baker, who designed some engineered proteins. We use those as other types of cargoes here, in addition to things like engineered transcription factors, to probe whether this engineered protein lipid interaction was useful for getting us that kind of balance between efficient loading but release in the recipient cells. And we showed that it could be useful for doing things like that. So I'll leave that for further discussion if you want to, but it's a neat frontier that I think has a lot to gain from understanding how this process works in natural EV biology as well. And of course, just zooming out a touch, this is now one of like a lot of related frontiers that are happening in this kind of delivery space. And I'm just highlighting a couple of ways in which defining the EV in a big tent way helps you see the connections between all these things that are happening here. So for example, there are these various different virus-like particles, including some that are derived from mammalian or human genome encoded archaic retroviral components, are shown to be able to do this kind of assembly, some loading things and delivery. It's a really neat frontier happening now, I think primed for the kind of applications we talked about, and maybe begging the kind of characterizations we need, because now we don't have no options, we have many options. And so if you're trying to build a thing, what you really want to be able to do is compare and contrast and figure out where the opportunity is, how to use the best part that's available from the scientific literature, and so on. So I'll pivot here to the second part of the story, which is the paper that I think inspired my invitation here, and just kind of talk you through briefly this one technology, and then we can end that with some discussion here. So this is bioengineering for the purpose of characterization specifically. And for us, a big motivation was what I just told you about. And I think with that, the convergence of the recognition that single EV characterization was just revealing all kinds of facets of EV biology that were previously either unattainable or unappreciated. And we wanted to think about how to connect that to some of the questions I posed. And just as an example, using things like antibody labeling of EVs together with high resolution microscopy lets you quantify things like the distribution of markers on EVs, the co-distribution of markers on EVs, and at the international meeting, ICEV, the use of data sets generated from instruments like ExoView and now FCM have grown exponentially, I believe. I'm making that up, but it seems like they've grown exponentially over the past couple of years. And so that's great. And I think the power is really well established. The instruments are generally usable, so we can compare across different experiments and labs really nicely. But one of the limitations is, and this is what kind of stuck us when we were thinking about using these for the previously proposed questions, most of these measurements are still relative rather than absolute, meaning you can say there's twice as many in this sample versus this sample, even twice as many per EV, but you can't really say there are 100 per EV, right? So that absolute threshold is something I'll dig into here. And really, one reason for that is that antibody labeling tells you how many antibodies are on the vesicle, not how many targets of the antibody are on the vesicle, because it's the same as for cells. There's a limit to which you can label things with antibodies. And so we got really interested in using these increasingly well available methodologies for doing labeling that can potentially overcome those limitations of affinity reagent mediated labeling. So namely, these are these self-labeling enzyme tags, halo tag, snap tag, clip tag. Those are the ones highlighted in this review article here. But the real power of these is a couple of things. One, the way these work is the tag itself is an enzyme, and the ligand you feed in has a synthetic substrate that these engineered enzymes act upon, and they kind of stop halfway through their normal enzymatic process, with a intermediate state being where they pause and a covalent coupling has occurred between the ligand and the protein. So in practice, that means you can add the ligand in, and it becomes covalently, irreversibly, effectively bound to that protein target here. And that's nice in a way because of this irreversibility I said, but also because these ligands can be made through synthetic chemistry, you can put all kinds of nice things on the outside. So in the most basic sense, you can use an Alexa dye instead of a fluorescent protein, which has all the benefits of some of those dyes in terms of photostability, spectral properties, and whatnot. And this review article is highlighting that that's not limited to optical microscopy. You can even use these for things like EM if you want to be able to tag them with things that are hard for the cell making the vesicle in our case to make. So that sounded really cool, and we decided just to see if this could work. And so this is the paper that I'll talk through for the next maybe 15 minutes and then jump into some questions. But it came out earlier this year, and it's just that. It's trying to use one of these tags, in this case the halo tag, to enable us to answer some of these single vesicle quantification questions. So the basic way we made this is similar to what was shown in that review article. We engineered our cells to produce a halo tag on the surface of the vesicles. This is, for the moment, the same exact display strategy we used to put antibodies on the surface. That's what prepared us to be able to do this. So we swapped out the halo tag, did a little bit of work to see how that works, and I'll show you that. But the scheme here is that we make the engineering stably in the cells, purify the vesicles have the halo tag on the outside. Once you've done that, you can functionalize those vesicles any way you want with these halo tag ligands. So for example, a dye can be added covalently to the surface of the vesicles. And the hope is that that will enable you to count exactly how many halo tags, the blue pack men or pack people, that's what we're trying to be able to count in this example. So first couple of things, we did our initial sweeps just to make sure that nothing strange was happening. So following MySEV guidelines, and also just in general making sure that there's a good check that nothing weird is happening. It didn't seem to have any big effect on the markers. If you look at the tag itself, we could find it in both the microvesicle and exosome fractions defined by centrifugation as before. And when we looked at the size distribution by NTA, there wasn't any notable effect on the size of the particles by expressing this particular engineered protein. Very similar to what we saw with the antibody display systems. Okay, so you can then start looking at this kind of fluorescence in a couple of ways. So first, this is just conventional flow cytometry, and we're going to use a couple of different instruments throughout this story, so I'll make note of which instrument's being used in each case. First, we're just using conventional flow cytometry to look at the halo tag expressing cells. And so you can see when you add the ligand, there's this ginormous shift in the fluorescence on the cells, which is primarily the cell surface, maybe a little bit on the endosome, given the way the ligand distributes. And if you look then at the EVs produced from such a material and do a bulk analysis, so for example, binding those EVs onto a bead and then running the bead through flow cytometry, you can see that qualitatively, and I guess to some extent, both quantitatively, there's a big boost in the vesicle-associated fluorescence in the samples where you would expect there to be. Okay, so can believe the fluorescence is attributable to the tag and to the ligand. That's all we got to so far. Then we went to town trying to see if we could answer some of these single EV questions. So the first instrument we used was an apogee instrument, and the apogee has the one we used has a single fluorescent channel, but it enables us to quantify things at a single vesicle level. And so when you look then at these different particles, and let's just talk through what these are. So these are unmodified EVs, meaning there's no halo tag genetically encoded, or the ones expressing the tag, and then we incubate them throughout the ligand, wash them a couple times, and run them by the instrument over here, the apogee. We can see that there is a large shift in the fluorescence associated with these. This was, by the way, the first time we had any insight into what the distribution looks like on the surface of these vesicles. So that's a neat way of looking at it. You can see there is a fairly uniform shift in the way this goes. This would be, not unlike the pattern we see over here at the cellular level, which is neat. And it seems kind of similar across the microvesicles and the exosomes. And if you look then at all these different conditions here, we can actually start getting at some of the numbers we care about. And I'll summarize those with some words here. First of all, we found that routinely over a number of different trials, 90% or more of the EVs are halo tag positive, fall above that threshold for the negatives. And if you then run a standard in your flow, which we can do, this is a calibration standard for the magnitude of fluorescence, you can actually count how many molecules of Alexa 488 dye are present per EV. And so that was the first time we had this number, which was, we had a bet in my lab. I think the bets ranged from 10 to 75 or something like that. And the answer was 1,500. And so that seemed wildly large. And so we did it a bunch of times, but it actually was pretty consistent in terms of like the number of counts we got through this. And if you do a little bit of math on the size of the halo tag ligand, or halo tag itself, which is a protein, this is the majority of the available surface area on the surface of the vesicles is covered by this tag. And now this is, again, a highly efficient display system. We're trying to push its limits. I wouldn't expect most natural displays are this magnitude, but if you push it, this is how far you could push it. And so we wanted to make sure those numbers were reasonable. So we looked at trying to confirm this by a few different orthogonal methodologies. First, we used same labeling system, but now we're counting the number of fluorophores using a high-res microscopy methodology. And the way this works is pretty neat. You can track the fluorescence of these particles over time, and as you extinguish the fluorophore, it notches down in intensity over time. And so you can count the number of fluorophores that were on that particle at the beginning of the photo extinction series. And so then you can generate a distribution like you would from a single vesicle flow experiment. Here you're seeing the distributions on exosomes and microvesicles by microscopy versus by this single vesicle flow cytometry. And this looks pretty similar here. If we compare that to a calibration curve, you can also do this conversion like you do by microscopy, and even just with total fluorescence over here, you can then come up with a quantitation of how many fluorophores were there. And so what we showed here was that for the exosomes and the microvesicles, still a large number of tags. It's 1,000. It's not 1,500, but within the limits of the methodologies, we think those are fairly consistent in terms of what the display efficiencies are. It's not clear which direction reality falls within that window, but it gives us a really good idea of how many particles are on the surface of each one of these vesicles. That was cool. And then we finally, just to kind of show that this was useful for other things, we made some, with a collaborator, some customized gold nanoparticles with the ligand, and showed that you can tag those. And when you wash them, you can visualize the number of immunogold, for example, like labels. These are not immunogold because it's a halo tag, but you can see more of these gold nanoparticles where the vesicles are, if they bear the halo tag versus the way it looks with the unmodified. And again, any TEM image, you should just say, this is an example of what we saw with the big giant grid. This is just to confirm that functionally it does seem to be doing what it should do. Not well suited to quantitation, I would say. It's much harder to quantify with this methodology, but qualitatively confirming what we think is happening. So last thing we did is actually look at some of these questions about co-display, and this will be the last little arc here. So one thing that's neat is you can buy these halo ligands with all different kinds of functionalizations. So first we bought two different Alexa4 dyes, 488 and 660, and we just mixed those different ligands in at different ratios with the coupling. And so first with the apogee, what you can see is if you vary the ratio of the fluorophores, you get a different, really linearly varying correlation in terms of the numbers of, let's say, one of the dyes, 48. Apogee is a single color instrument. So what we can see is you can see the competition really between the ligands as you mix in the competing fluorophore dye. So that's great. And it does show that it's titratable, but we really wanted to know whether these cartoons right here are what they actually look like. And so we, core facility here obtained a nano FCM instrument around this time in the process. And we then were able to run these two color versions of that experiment. And so looking at this either from a quantitation per particle or kind of a conventional biaxial flow cytometry plot, you can see that co-labeling is evident in these particles here. So this is just one ratio. You can clearly see the double positive vesicles here. The controls are single positive as you'd expect. And this titration we observed in the sort of competition way, you can actually see it in terms of the Alexa 48 dye going down and the Alexa 660 going up in those same corresponding directions here. So it's neat, and this really means that you can make one batch of EVs and try a bunch of different ways of co-display. So for here, we're doing quantitation, but of course, this resonates very closely with what we talked about in the first bit, because if you wanted to explore different things that might have a bioactivity, for example, you don't have to do another genetic engineering approach. You can just buy recombinant versions of what you want to put out, tag them on in different combinations and levels, and if it works similarly to this, you can do some experiments with some rapidity, I think. The last thing I'll say before I leave is that these numbers, which I think are really, go back one slide here. These numbers are really provocatively large, I think. That's the way we thought about it. We were gratified to see some really neat work coming out of the team at Kodiak with a different system that showed that indeed they were getting very similar numbers in terms of how many proteins they could display on the surface of the system with their display methodologies. So the last thing that we wanted to do then was kind of look at how these halo tag methods provide new insights into those kind of cross comparisons across labs or across technologies here. So in this last experiment, we looked at comparing the scaffold we had been using, this PDGFR, with the one that Kodiak had used and characterized in their experiments. And they reported this level of labeling that was concordant, but a little bit different system. So we wanted to do a direct comparison. And what you can see here is if you look at a couple of different versions here, this is a short and long truncation of this particular display scaffold. And for the sake of this, we're really just comparing a couple of different design scaffolds to see what happens. And this just shows how this halo tag makes that really easy to see a lot of cool stuff. So for example, you can see that both of the display scaffolds give you labeling on both exosomes and microvesicles. The distributions are quite different in the sense that the PDGFR gives you this kind of peak separation here with the labeling, whereas the PTGFRN long gives you more of a continuous distribution. That may be two overlapping bits, but in any case, you can pose those hypotheses. Big difference between the two here. And I think what's interesting then is if we looked then at the mean particles per or fluorophores per particle, we do see comparability, especially in the microvesicles. These two different scaffolds give you maybe a similarly high labeling efficiency. But if you look down here, the distribution is different than you might infer if you just imagined these were the same distribution, but with different means there. So I guess the point is there's a lot of different information you can glean from single vesicle characterizations like this. And for our sake, I think this just tells you how, if you have a lot of different choices to make, you want to make them in a quantitative and unambiguous way. Halo tag is really handy as a thing for that. And I would suspect the other systems that work in this way could be similarly extended to do this kind of nice functionalization. That's a speculation. So I'll end the talk with just a couple of comments. This is a artistic rendering of what we feel like EV bioengineering has become, which is we have, like, of course, genetic tools for doing what, when you say engineering EVs, I think many people outside of the scientific background, think about tiny robots putting together these nanoparticles. And of course, that's not how it really works. We're using genetically programmed self-assembly. But the fun thing is it's becoming more like this. We have enough tools that you can make choices as to how you want these particles to self-assemble. We have ways of actually driving that self-assembly. So the last couple comments I'll make is that that's a big part of why we decided to spin this out into the company Cyanix. So I'm just raising this in case folks want to hear about it. But the point was, we realized at this point, this is technology that could be used for a lot of applications that exist today in the form of making better delivery vehicles. And that tech set I just told you about is what's now been commercialized in a startup company. I want to thank my lab. The folks who contributed to this specific arc of work are the ones named here, with particular shout outs to Dr. Michelle Hung, Dr. Devin Stranford, Dr. Taylor Gunnels, and Dr. Roxy Mitrud, who led a lot of the EV bioengineering work I showed you about today. And if you like this approach in general, but a blatant advertisement, there's a lot of cool synthetic biology happening at Northwestern. We would really encourage folks interested in doing undergrad, grad school postdocs with synthetic biology labs. I'm happy to connect you with some of those folks. And I will leave this picture up here and take any questions anybody might have. Thanks a lot.
Thank you very much, Josh. It was a super interesting talk, and I'm really excited to see what the future holds for EV engineering in the future. So we have some questions. I'm going to be a bit selfish and start with my own. So I had some questions about the active loading method. Have you tried using this method using particular proteins or motifs that are targeted towards specific subcellular regions of the cell, maybe an organelle, like the ER or something?
We have not. The closest to that is with the lipid story that I mentioned, just in passing. We looked at different tags, using tags that can encumber different lipid modifications. And what we did in that story was look at sort of how the different transmembrane domains and, say, lipid modification domains that you put on your engineered protein interact with the different features of different cellular membranes. And so, for example, we did a lot of comparisons between raft-like parts of the membrane and non-raft, which of course are transient and whatnot, but you can still do some of these analyses. And so we can definitely partition to those kind of different, say, membrane biophysical domains using those protein lipid interactions. We've not yet extended that to something like you're describing, like looking at a specific, say, interaction with a protein that already has a natural location at one of those different compartments. But I do think that's a natural extension that you could imagine trying, right?
And just to add to that, have you ever tried using drugs, specific EV inhibitors that are currently on the market, like the one against enzymes 2 and manuisin A, and whether or not they would, what impact they would have on the active loading tool that you've used?
That's interesting. We have not looked at that. We did some work that I don't think ended up in the publication, looking at back when we were trying to figure out the limits of delivery, and we mentioned fusion we thought was the limit of delivery. We did try playing with some endosome disruptors. So it's the other direction, really, of this. And didn't see a big effect when you add some of these things in. Like once you have the assay that we were using, and that's probably limited to T cells in this case, we didn't see a big boost, for example, in the degree to which we could get a functional readout or when added in combination with the vesicles, or maybe in place of the viral proteins. That was kind of our thinking on that one too. That's a very limited scan that we did. Yeah, but the NSMase inhibitors is interesting in part because that would, those are the sphingomyelinase synthesis pathway proteins, right? And so that should arguably have an impact on the distribution of lipid compositions on the different membranes. So I would imagine that kind of an approach could be interesting for interacting with some of those things. We played a little bit with that, but not in the exact direction you're asking about here.
Okay, so I'm going to start with some questions from the audience. Antonin Marquand asks, "Thank you very much for this amazing talk. What are the advantages of using the SNEV as a targeting motif as compared to other IgG derivatives, according to you?"
Oh, yeah, I don't know that there is one. In fact, I think there probably are. Well, yeah, so let me just generalize that. I don't think I would pitch the SCFE as the best domain for doing this. So there are a broad class of affinity reagents you can imagine using here. The big jump for us was peptide to SCFE. And so the challenge with peptides, there's an exception where there's a few peptides that work really well. We did some work, for example, looking at some of these ones that happen to work really well. But we did some other studies showing that other peptides that have some effect don't really marginally impact the way the EV delivery occurs, just because the nature of that affinity, maybe the entropic penalty of binding and whatnot. And so there is a much stronger affinity you get when using an SCFE compared to most peptides that people have used. And so that was the big attractor for us to go to SCFVs. That said, if you're comparing SCFVs to nanobodies, to DARPins, to things that are affibodies, I think actually you get that benefit with that whole big class of big proteins. The big leap for us was we had done some previous work looking at what happens when you put peptides in the surface, actually inspired by Matthew's work. And a big challenge when these are deriving from the exosome compartment is that the proteolytic activity destroys a lot of those ectodomains on engineered proteins that have maybe not evolved. Like LAMP proteins have evolved to survive that process, but a lot of the engineered ones haven't, and so they get degraded by proteases. I think then that was our big concern with the SCFVs, is that these are big proteins that haven't evolved to be in that display system. Will they be chewed up as well? And so doing the work we did, we seem to have overcome that. And I think if you were to pick a new target, that would be my caution to you, is that I imagine that's something you want to still be aware of. It may be that it works. That's something that I think just needs to be checked every time. And certainly there's a different risk with the microvesicles or the surface budded biogenesis pathway versus the endosomal, route of EV biogenesis that make exosomes. So I think there's probably a way to do it for all those different scenarios, but that's what I look for. I think the other thing, folding is also something we think is probably a challenge. So even though we made our own SCFE from a published antibody sequence in this, we had to play with a couple of different ways of doing that. There are probably fewer hurdles to climb if you're doing a single domain like a VHH or an antibody domain, just because they don't have that potential for folding errors in the same way that SCFVs do, but both can work.
Yeah, and just to follow on about the folding, did you have any stability issues when working with these EVs, which are already naturally quite sticky, along with these variable chains?
Yeah, we didn't, in terms of stability post, like after you harvest the EVs, we didn't notice any problems. So we did a bit of checking on that with looking at storage in the fridge, freeze thaw, things like that. No, I mean, I think this falls into the category of the vesicles themselves are very stable, right? And generally don't seem to have aggregation issues when they're just sitting around, for example. And nothing we did change that. They seemed like they retained the ability to, for example, if we did like an uptake assay after having stored it in the freezer or freeze thawed a couple of times, you get roughly the same behavior after a couple of different rounds of that, which is not what we expect if it were being disrupted.
Okay, so next question is from Claudia Velena. She asks, thanks for this awesome talk. What type of high-res, super-res microscopy did you use to look at your fluorescent EVs?
Yeah, so this was actually, the picture I showed you is the instrument we use. It's an Olympus instrument. Okay. So this is done by a collaborator who actually does a ton of work. Mitchell Wang's group does a ton of work using these high-end microscopy things to do different types of science. In general, they do a lot of really neat materials characterization where, for example, they're trying to track fluorophores inside of polymer matrices. And so we use the setup they use for that in this case. I think actually we did try to write in the paper what you would need to do that. I think that kind of setup is a bit overpowered. I mean, colloquially overpowered in the sense that like it probably can be done with different instruments that don't have necessarily those capabilities. You just need to be able to attribute the fluorescence to individual things you've deposited. So if you've got a nice confocal, you can use perturb things like that, then I think there might be some ways of adapting a methodology to do that as well.
The next question is from Dr. Natalie Turner. She asks, do you have any plans for in vivo experiments? And if so, what are they?
Yeah, so I didn't go down that road, but yes, we do. And we've done in vivo experiments, I guess. I'll tell you the questions we're looking at. So on the translational side, using these to deliver genetic cargoes is something we're very interested in pursuing in vivo. And it's partly what we ended up spinning out to the company so they could pursue that in different routes. But just thinking about how to make delivery in vivo specific and efficient is certainly a big ongoing area of work. We have some ongoing stuff in the lab focusing on that front as well. And also just questions that are to do with things like circulation time, bioengineering, distribution, things like that. So I think we're still pretty heavily focused on the enabling technology component in the group rather than going deep on a specific therapeutic model at the moment. More by choice, that's kind of where we've chosen to make our contributions on this. But once you can do a stable gene editing type of application, it's kind of ready for use in a disease-specific model there. So I'd say not guaranteed that it will work or whatnot, but the next relevant question becomes, is this degree of genetic editing adequate for this, animal model, for example, to show a therapeutic response? That's kind of how we have structured that. Yeah, I guess I'll leave it there. Happy to follow up if that wasn't adequate.
So the next question kind of follows on from where you just finished, actually. What's your take on engineering EVs for interspecies communication or maybe inter-kingdom? Do you see it as possible, and do you think it'll be useful for the future?
That's cool. That's actually one of the things that got me hooked on this in the first place, was looking at, so of course there are like every cell makes vesicles, right? And so the potential interaction between things like gut microbes and the vesicles they secrete, outer membrane vesicles or whatnot, and the intersection with sort of host cells, that's super exciting. Seems to be some really neat evidence that that's part of what's happening in that ecosystem. So that's very exciting if you imagine engineering on the microbial side. We're trying to do delivery on the human side in this case, or I guess animal side if you wanted to do a veterinary application. Tons of cool stuff to do there, I think. And yeah, interkingdom in general. I mean, the other thing that's very interesting is I think it's still a pretty big open question as to how these vesicles play a role in natural interspecies communication. Again, I'm thinking gut microbes, but maybe just in sort of that kind of microbial consortium setup. I will say EVs keep popping up in other stuff we do in the lab too. So for example, we have a story where we're trying to understand how immune cells make decisions. And it ended up coming down to an observation that we discovered a new facet of the way that macrophages seem to communicate with each other. And it is plausibly an EV-mediated route that is not like direct cytokine signaling, something else that involves EVs. So I think there's a ton of evidence, maybe in addition to the very well explored parts of literature, that this kind of stuff can play a role. And again, the way I would answer that question is, I think this approach of trying to engineer it to understand it is super fruitful, because if you're looking for EV phenomena, of course, we all know this, a big challenge is it's a slow burn, right? EVs are made in relatively low numbers that have an effect over very long time scales and in very subtle ways, and those are extremely hard to study, right? So these observational approaches can be benefited by pairing up with something that's more, like I said, not just what do they do, but what can they do? And so if you can engineer a microbe to deliver to a macrophage, that might be useful for an application standpoint. It also may be a clue that that's what's happening naturally, but of course that's an artificial system. And so it gives you though a starting point as to what to look for. So I think that's where I'm really excited about thinking about these tools as really ways of perturbing the natural systems to figure out where those opportunities might lie.
Okay, so next couple of questions are more about the second half of the talk. So first one's from Carlos Jesus. He says, what's an amazing talk. Have you tried to use this halo tag technology to load biomolecules such as genetic material and proteins? And have you tested whether or not they're still functional after delivery?
Have we used them to load proteins? Well, we haven't done the thing that I said I think would be cool, which is, I mean, we contemplated this in the paper and whatnot, but the fluorescent ligand that we added to the surface, you could substitute that with an antibody, for example, right? And then use that to functionalize the surface. I think the trick is the way the halo tag works right now is it's at least partly on the surface. This is an important thing. It's actually very hard to know if it's entirely on the surface or both on the surface and in the interior. We can use non-membrane permeable ligands to know that it is at least on the surface, but it doesn't mean that it's not on the interior as well. So depending on what cargo you're trying to load, you may want it to be on the interior. It may be fine for it to be on the exterior for other types of cargo. If it can be on the surface, then I think it's ready to go with what the technology is. If it needs to be on the interior, you might need to combine the surface functionalization with something that's either loaded before or after biogenesis to get it onto the interior. And we haven't done a ton of that with Halo.
Okay, go ahead, Ayush.
So you talked about cargo loading by targeting the lipid rafts for increasing loading. Have you also looked at increasing uptake by targeting these lipid rafts? Like I'm talking about lipid rafts are also the sites for endocytosis. So
Yeah.
maybe targeting those sites in the target cell might also improve uptake. Have you kind of looked at that?
It's a good idea. So we haven't done that directly, but I think you're absolutely right. In fact, there's, two ways to think about that. We know, let's take a page from the targeted delivery of viral vectors and nanoparticles, right? We know there's a big difference in targeting target 1 and target 2 on a given cell, even if they're expressed at similar levels. Few reasons. Maybe there's an endocytosis effect. The other might just be membrane compatibility, right? And so for those receptors that tend to partition into different membrane subdomains, they may be more, say, facultative for mediating that eventual membrane fusion with your vesicle or
Yes.
envelope viral vector because of that aspect of membrane compatibility. It's certainly speculative at this point, I would say, but I think it's a really good idea. This is my colleague Neha Kamat, who's behind the wall right now here at Northwestern. That's a really interesting thing that they're excited to explore. So we're definitely going down the road of seeing how we could maybe gain additional biophysical insights into how the rules governing fusion might be a way of harnessing that kind of an insight. So nothing yet, but I do think it's a really good question.
Yeah, thank you.
Okay, so I'm going to leave it for the last two questions now. So my next one's from Jack Mellon. He says, thanks for the fantastic talk. Do you have any thoughts or experiences with microRNA delivery by EVs? They're an early career researcher and they're trying to figure out the best ways to deliver microRNAs locally.
We haven't looked at that a ton. So here's the closest answer. One of the stories that we worked on at the beginning of our entry into the EV field was trying to enhance RNA loading into vesicles. So we used a system we built a system that uses the MS2 loops to drag RNAs into the vesicles, and did a lot of stuff to show how that works and things like that. There's been some really neat subsequent work from a different team from Smirel and Lucy's group showing how that concept can be, I think, better implemented with some new domains and things like that, where they actually can show some neat RNA loading and functional delivery. So that's the closest thing I've seen to an active loading of RNA, this kind of in general, RNA binding domain binds to your RNA of interest, and that causes it to be loaded into the vesicle in a certain way. And that is the setup we've used. Like I said, we didn't use microRNA, or I didn't say, but we didn't use microRNAs as a target. We were actually trying to see what the rules were for how big the RNA could be before you start running into problems. And the real issue is you first discover that most RNAs are just chewed up in the cell. And when you look for that, you find that a lot of the chewed RNAs end up being the ones you're loading into the vesicles. You get some of the full-length ones, but there's a lot of fragments that are in there too. So the degree to which you're working with a stable, small RNA structure, I think it's useful. The one thing I would say is we saw much more of a marginal benefit for larger RNA cargos compared to when you have small RNA cargos. For small RNA cargos, which would fall into the microRNA range, just abundance is probably enough to get you a pretty good amount of loading into those vesicles based upon the sizes we saw, for example. So that's my bet. It's not guaranteed, and it probably has to do with the way those microRNA are being processed in the producer cell as well, depending on which stage of processing you want them to be loaded into your particles. There's some neat work from Tom Schmidt's group and a few others who've looked into that question. If you haven't read their work, more detailed, yeah.
Okay, and just final questions from me. So in a few of your slides where you were characterizing the halo tag EVs, one of the things you showed was the number of fluorescent ligands between the exosomes and the microvesicles. And they looked very similar despite the size differences and the site of biogenesis for these two different EV subtypes. Do you think your microvesicle size distribution is a lot more closer to EVs? Maybe they range from 100 to maybe 500 and not the crazy 1 micron that we usually see?
Yeah, that's a great point. That is 100% true. It is the case. The size distribution is very similar with the vesicles that come from the HEK293s that we're using in this case. So if you looked at that, I guess I showed one NTA analysis that kind of shows you those distributions. But if you quantify it, even there's some difference, but they are more overlapping when we look at using the same methodologies and vesicles produced by those cells compared to the similar density gradient methodologies applied to other cells. And frankly, if you use other methodologies to separate those fractions, the answer is still the same. The size distributions are much more similar for these HEK-derived vesicles than are probably the case for those that come from other cell types.
And are you currently testing other cell types, or are you just going to continue kind of further developing this technology for other things?
Well, yeah, we have other more wild things that are probably not ready for YouTube that we're looking at. But yeah, we are looking at vesicles from other types of cells. And I mean, there's no spoiler on this. I mentioned we were thinking about EVs as like a force multiplier. You can make a really powerful cell therapy if your cell that goes into the body produces therapeutic EVs as a product. And so certainly then whatever therapeutic cell you're talking about, its identity and EV distributions will start to touch on some of these questions about loading frequencies.
Okay, so I think that's all the questions. Once again, I want to say thank you again to Josh Leonard for your time and amazing presentation. To everyone in the audience, just before you leave, just to give you a heads up that this talk was recorded and it will be on our YouTube page in a few days. So please look out for that. So let's go to YouTube, type in SNEV Research, you should be able to find it there. You will also be able to find all of our previous recorded talks as well, in case you missed those. And also don't forget to sign up. It's completely free and you'll get notifications about talks like these in the future.
Could I jump in and just say thank you to everyone who came today? Fantastic questions. Thank you all for the organizers for inviting me today. Look forward to following your series as it goes forward.
Thank you so much.
Thank you very much.
Have a great day.